How does epigenetics affect osteoarthritis genetic risk? Imagine your genes are like a set of instructions for building your body. Now think of epigenetics as the switches that turn these instructions on or off. These switches don't change the instructions themselves but influence how they are used. When it comes to osteoarthritis, these switches can make a big difference. Osteoarthritis is a joint disease where cartilage wears down over time. Your genes can make you more prone to this, but epigenetics adds another layer. It can change how genes related to joint health are expressed without altering the actual DNA. These changes are often influenced by your environment and lifestyle. For example, aging, injuries, or putting too much stress on your joints can trigger epigenetic modifications. One common epigenetic mechanism is called DNA methylation. This involves adding chemical groups to your DNA, which can turn off certain genes. In cartilage cells, increased DNA methylation can reduce the production of proteins that protect cartilage. At the same time, it can activate genes that promote cartilage breakdown. This imbalance accelerates joint damage seen in osteoarthritis. Another mechanism involves histone modifications. Histones are proteins around which DNA is wrapped. Changes to these proteins can make genes more or less accessible for activation. When histones are modified, it can influence the expression of genes important for maintaining healthy joints. Regulatory RNAs are also part of epigenetics. These small molecules can control gene activity by blocking or promoting the production of specific proteins. In osteoarthritis, regulatory RNAs can affect how cartilage cells respond to damage or aging. Genetic studies have shown that many risk variants for osteoarthritis are found in parts of the genome that do not code for proteins. Instead, they regulate gene activity. Epigenetics helps explain how these variants influence gene expression in joint tissues. Certain genetic variants can affect DNA methylation patterns, which then change how genes involved in cartilage health are turned on or off. This interaction between genetics and epigenetics means that even if you have a genetic predisposition, environmental factors can modify your risk. For example, a person with genetic risk factors might never develop osteoarthritis if their epigenetic patterns stay favorable. Conversely, adverse environmental influences can increase risk by changing gene expression. Understanding this relationship opens the door for new treatments. Since epigenetic changes are reversible, scientists are exploring drugs that can modify DNA methylation or histone modifications. These therapies could help slow down or even prevent cartilage deterioration. In addition, epigenetic markers might help identify people at higher risk for osteoarthritis early on. This could lead to earlier interventions, lifestyle changes, or targeted therapies to protect joint health. To sum up, epigenetics influences osteoarthritis risk by controlling how genes are expressed in joint tissues. It acts as a bridge between your inherited genetic makeup and your environment. This gene-environment interaction explains why some people develop osteoarthritis while others with similar genetics do not. It also offers promising possibilities for future treatments and prevention strategies.